Genetics, I hear you say? How's our understanding grown since Mendel and Darwin? And what has happened as a result? Well, genetics as a term was first coined by the English biologist William Bateson. He revitalised interest in Mendel's work and really initiated the scientific discipline of genetics. While he was doing this, Sir Roland Biffin was exploring the use of Russian wheat genetics at his laboratories in Cambridge, looking at how he could improve the wheat plant's resistance to yellow rust. This work resulted in the creation of the wheat variety known as Little Joss. As a result of this, in 1912, the Plant Breeding Institute was formed and Sir Roland became its first director. In the years that followed, scientists at PBI, as it became known, set about improving agriculture using sound scientific principles. Using genetics sourced from North America, they produced the variety Yeoman, which set the standard for wheat yield and quality right through to 1957. Now, just a short while after PBI opened, RAGT was set up by a group of farmers in southern France. They set out to improve agricultural seed varieties for farmers in the region. And in 2004, RAGT purchased the PBI cereals portfolio. Since the end of World War II, there have been a number of notable advances in plant breeding technology, such as the introduction of dwarfing genes from Chile, discovered by the American agronomist Norman Borlaug. Now, this allowed farmers to push varieties to produce higher yields with a reduced risk of the crop lodging and to reap significant economic benefits in the process. Let's not forget the development of single seed descent, which allowed the rapid cultivation of large numbers of plants, and this process was aided due to John Bingham's use of lit glass houses and the use of cold stores, which meant that not one but 2 generations of wheat could be grown each year. And importantly, the massive breakthroughs in DNA technology in the 1980s, which gave rise to DNA markers, allowing plant breeders to identify important variety traits. And on the subject of DNA markers, we neatly arrive here in the present day, where modern science and technologies are just allowing breeders to unlock genetic secrets hidden within different species and varieties of crops. Before genetic markers, wheat breeders had to go on phenotype alone, which is the physical expression of genes. For example, the number of tillers, or the shape of the wheat ear, or the presence or absence of disease. Take the example of orange wheat blossom midge resistance. Well, a breeder could see which plants had midge and which did not, so they knew which varieties were resistant. However, to arrive at these conclusions was a long and costly process. Mapping the wheat genome was a critical step forward in allowing breeders to more fully utilise genetic markers. And believe me, it's helped turn agriculture on its head. Mapping the wheat genome was no easy task. It took 13 years to fully map the human genome, and the wheat genome is, well, wait for it, nearly 6 times bigger. Okay, stick with me here because I'm going to give you a quick explanation and overview to genome mapping and markers. Now, the term mapping is exactly just that, basically mapping the genome to provide breeders with a greater understanding. Let's take the London Underground for example. Without a tube map, the whole network of tunnels and stations, it's a pretty mind-boggling place, but with a tube map, we can start to understand how it's all connected and where in London certain tube stations are situated. Well, the same applies to a wheat genome. Once we start mapping it, we can start finding out where certain genes are located within the whole structure. Let's keep with the London Underground theme to help explain how breeders use these markers. Again, the clue is in the title. Just as we would place a mark on the tube map to show the location of a station, we place a marker on a string of DNA to highlight locations that are responsible for certain traits like orange blossom midge. As breeders started to really get to grips with mapping and markers, they started to realise that they could identify the location of one gene by the presence of another. It was a big job and it took a long time, but as a result, our understanding of wheat plants and their resistance to pests and diseases has grown massively. So, in the case of orange wheat blossom midge resistance, by comparing the genes of resistant and non-resistant varieties, the breeder could identify the marker associated with the gene, which we now know as SM1, that was giving rise to the resistance. To illustrate this, here's a small section of the tube map with the stations Holborn, Covent Garden and Leicester Square. Holborn is marker 1, Covent Garden marker 2 and Leicester Square marker 3. We believe that there's a cinema near Leicester Square and we want to go to it. In this way, we discount Holborn and Covent Garden and concentrate our search around the Leicester Square marker 3. Now bear in mind, this was the early 2000s and DNA marker technology was still in its infancy with little in the way of throughput. The discovery of a marker for SM1 enabled breeders to track the orange wheat blossom midge resistance gene in their programs, knowing that in most cases, if the marker was there, then the gene would be there too. Once we've placed a marker in a rough area of where a certain gene might be, in this case Leicester Square, we then need to map around this area and place further markers until we narrow down the location of the actual gene we're looking for. That's orange blossom midge and not a cinema. This method is now possible through further advances in DNA technology, meaning more markers can be used to identify genes' location. So in the case of orange wheat blossom midge, we now have the markers on either side of the gene, which greatly increases the accuracy of selection. Of course, the ultimate aim is to identify the gene itself and a marker within it to ensure 100% accuracy. As well as improving accuracy, better technology enabled increased throughput and data analysis. In the RAGT lab alone, there was a 5-fold increase in the number of samples analysed between 2002 and 2018. Now, marker analysis is also vital for identifying the absence of a gene and therefore the likely absence of a particular characteristic. This allows us to reduce the number of lines we're subjecting to physical tests, and that saves time and money. Again, let's go back to our London Underground map and treat the stations as genes. For some traits, such as orange wheat blossom midge, it's a single gene that confirms resistance, but it's seldom that simple. Resistance to yellow rust, brown rust, septoria, and many viruses involves a number of genes, so it's much more complex to track. Traits such as yield and vigour are an even bigger nut to crack, involving multiple genes randomly spread out. Understanding what those genes are and how they interact is crucial if we want to improve yield and stability of the plant. Another example of how DNA technology helps breeders is genomic selection. This allows us to get a snapshot of the whole genome, which we can use to better understand complex traits such as yield. It enhances the information we get from the field in a given year, and this in turn increases confidence in the yield potential and the stability of a particular variety.